Thank you so much. I just assumed that I would speak after the coffee break. So I want to thank the organizers for inviting me today. It's been a fantastic 2 days already, and I'm really looking forward to the rest of this meeting. And I'm going to talk about something a little bit different. So I'm going to ask you to quickly reprogram your adult brains. Um, but I think it's still really pertinent to everything that we've been speaking about so far. So I'm interested in how behavior is regulated, and really from sort of a gene regulatory perspective. And so within that, we're interested in how the environment regulates behavior and where individual differences come from in behavior. We're also interested in pleiotropy and how pleiotropy, thank you, plays into this. But I'm not going to talk about pleiotropy today, but Marla actually, I think, will talk a little bit about it. So I don't need to tell you, we've talked about this a lot, but usually we think about behavior coming sort of from an interaction of nature and nurture. So a part of it is reprogrammed in our genes, and a part of it comes through environmental influences, and these two things interact with each other. And it's not a simple interaction. In fact, it's often very complex. And from the perspective of the environment, we often think about epigenetics being the process that mediates that environmental influence in biology and behavior. And I'm not going to go too much into epigenetics, you're all very familiar with it, but basically it changes chemical tags on histones and on the DNA that affects gene expression in a lasting fashion. And today I'm going to talk about an added layer of gene regulation, which has come to our attention relatively recently, so in the last 10 years or so. It's been rediscovered. It was actually discovered in the 70s, but then it lay dormant for a really long time. And now we're rediscovering it, which is epitranscriptomics, which is just like epigenetics, the addition of chemical tags, and there is actually over 125 different tags that occur on RNA and not on DNA. And they've been pretty well studied on non-coding RNAs, such as tRNAs or rRNAs, which we've talked about today. And they're really important, these modifications, for the 3D structure of these RNAs in the cell, which allows them to accomplish their functions. But more recently, we've discovered that they not only occur on these non-coding RNAs, they also occur on mRNA, so the RNA that encodes for proteins. And we don't really know very well what they do on mRNA. And because it is always difficult to study causation, we're approaching this question in how does epitranscriptomics regulate behavior, and where does it play in this relationship between genes and the environment, nature versus nurture, in a model organism, which is the fruit fly, in particular because it is very easy to manipulate both the genetic and the environmental component in fruit flies, but many processes are very conserved across animals that occur in flies, so we can study some of the basic genetic processes and infer quite a bit. So a little bit more about mRNA modifications. There's generally two separate classes of them that we think about, and this on top right here is a linear representation of an mRNA transcript. We have a structure that occurs in every mRNA transcript, which is called the cap, and then you have an untranslated region, which is regulatory, then you have the actual coding sequence of an mRNA, and then an untranslated region, which is also regulatory, the 3' UTR, and then a poly A tail. And the cap is heavily modified, and it's heavily modified in all transcripts. And we thought that that cap modification is important for animals to recognize RNA transcripts as their own and to not degrade them. So we know that if we remove the methylated cap, transcripts are degraded, and so for a long time, people thought it's mostly there for stability of the transcript and for self-recognition, that it's internal RNA. However, there is some evidence showing now that it might be important for other things as well, such as subcellular localization of RNAs. So there's some indication that the cap is important in moving RNAs in neurons from the cell body to the extremities of the neuron, where it can be important for neuronal transmission. And in the gene body itself, there is a variety of different modifications that can occur. And just to highlight here, there's different modifications that occur on all nucleotides. So adenosine, cytidine, guanosine, and uridine, they're all modified, and including the sugar bone of the mRNA. So the ribose can also be methylated. And we're really just at the start of trying to figure out what does each of these modifications do, where do they occur, and how do they interact with each other, and ultimately what is the fate of a transcript that is modified. But today I'm only going to speak about the most common mRNA modification, which is M6A, or N6-methyladenosine. So we know that this modification occurs in about 0.5% of all adenosines in a transcriptome is modified, and this is the highest modification for mRNA that we know of all the modifications. So they're not that high, but this still translates to several thousand transcripts within a cell that carry the modification. And so M6A is also the most widely studied mRNA modification, and we know that it is deposited on mRNA through a complex of proteins, which we call the Anreiter proteins. The central protein here is metal 3, which is the enzyme that deposits the modification on mRNA transcripts. And then there's a bunch of other enzymes that act as cofactors. I want to highlight one right here, which is FMR1, which you might all be familiar with because it causes Fragile X syndrome in humans. And in fact, in the schematic, this is a mistake because it's not a Anreiter protein. We know now that it is a protein that recognizes M6A in transcripts and is an effector protein. It's not a Anreiter protein. Then you have a set of proteins that remove M6A from transcripts that have been methylated, and these we call eraser proteins. And this indicates that this M6A modification is a dynamic modification. And we know that it is deposited on certain genes in response to stress, in response to the external and the internal environment of the cell, and that it is dynamic, which makes us think that it might have an important role in sort of the dynamic regulation of the response of the cell to a stimulus and of the individual perhaps to external circumstances. And lastly, there is a set of proteins which we call reader proteins, which FMR1 is one of them. And these proteins recognize the modification and then determine through the interaction of protein complexes what happens to that transcript that has been modified. So depending on which reader protein is recognizing the modification, it can affect translation, so the amount of protein that is produced from that transcript. It can affect decay or stabilization of that transcript, so how long-lived it is in a cell. It can affect splicing, and in fact, a lot of the sort of concrete examples that we have of function of M6A are related to splicing. And in fruit flies specifically, M6A acts through the splicing of one specific gene in sex determination and sex-specific phenotypes, such as courting behavior. And we also have some evidence that it can affect nuclear exports, so where the transcript is localized in a cell. All of this, of course, ultimately affects cell function. From a biological perspective, we did a little compilation of everything that we know of what M6A might be doing at a biological level, and there's quite a bit. So there are developmental phenotypes like embryonic development, meiosis and gametogenesis, stem cell differentiation and cancer. But there's also quite a few higher level behavioral phenotypes. It's involved in circadian rhythms, it's involved in flight and locomotion in flies, but also in food choice and reward and obesity and heart disease. And I want to point out, so around the top, I have all the different organisms where we have evidence for that it is involved. So it seems to be quite conserved. For instance, if I take If I take a look at circadian rhythms, it plays the same role or the conserved role in circadian rhythms in Drosophila, in plants, and in humans. So that is always nice to see for us Drosophila researchers because it indicates us that any findings that we have, we might be able to eventually translate that to other animals as well. And we are particularly interested in the behavioral phenotypes because we're looking for individual differences in behavior and how those are regulated. So here I want to point out circadian rhythms and food choice and reward, and a little bit of brain development, which I'm going to talk about today. So the use of model organisms, of course, that you can do genetic manipulations, and what we've done to start was to make a null mutant a functional mutant of this core enzyme in the complex, which is metal-3. In flies, it's also called inducer of meiosis 4. That's why it has a different name here. So we deleted methyl-3, and these flies don't have any M6A left. And as opposed to mammals, they survive without M6A. All mammalian model organisms, if you completely delete this gene, it's embryonical lethal. So at that point, we worried a little bit about the actual function of it, because how important can it be if it is not essential? And we know that Drosophila is not the best model organisms for some epigenetic studies because, for instance, flies lack all DNA methylation. They don't have any DNA methylation and seem to be fine without it. So we did a longevity experiment, and we looked at these metal-3 mutants, which are here in red, and compared them to wild type, which you can see in blue. And we see that even though they survive to adulthood, the mutants, they do have a very reduced lifespan. So it does seem to have an important function. And so if we look at that, if we plot the standardized median survival of these mutants compared to a wild-type control, we see about a 72% reduction in lifespan, in median lifespan. The interesting thing is that we had a transgenic rescue where we reintroduced a transgenic copy of the metal-3 gene into this mutant, but we only introduced one copy. And so usually you would have two in an organism, and these rescues only have one, so they have half the dose of metal-3. And these are the animals in yellow. And what kind of surprised us is that compared to wild type, we saw a 20% increase in lifespan. And this is a very rare thing, even in flies, that you can see an increase in lifespan with anything. And because there's been a lot of talk recently about how important sex differences are, we decided to do this not only in both sexes, but also in mated versus virgin animals, because we know that mating status has a very big effect on a lot of phenotypes, such as metabolism and behavior. And the interesting thing is that even though you see the effect of decreased lifespan in metal-3 mutants and increased lifespan in these rescue animals in both sexes and in both mating statuses, they are there, but to different extents. So in virgin males, the effect is much bigger than it is in mated males. And in fact, if we were to plot this like I did in the previous slide, this translates into a 45% increase in lifespan in these rescue animals compared to wild type. So if we just for fun were to translate that into human years, it means that all of a sudden our median lifespan would increase to 106 years, which is a huge effect. So then we were wondering, how could this increase in lifespan, or through which process could this increase of lifespan be mediated? And I forgot to mention, everything I'm showing to you today is unpublished data. It is all very new, so I don't have all the answers for you today, but hopefully soon. But we think that this effect of M6A on lifespan might be mediated through food intake and metabolic phenotypes. So we know that in yeast, RNAs that are methylated with M6A are enriched on translating ribosomes after vancomycin treatment. So it means that there is an increase in protein being made from those mRNAs after vancomycin treatment. And vancomycin targets a pathway, the TOR pathway, which, when it's activated, decreases food intake and body weight. And we also know that METL3 mutants are leaner as juveniles, and we have some preliminary data that they're also leaner as adults. And this seems to be happening through an interaction of M6A with the NAB2 protein, which in humans is called ZC3H14, which is also a model for intellectual disability in flies. And in humans, it's a heritable form of underlies a heritable form of intellectual disability. So I'm pointing this out because there seems to be this recurring link between brain development, neuronal phenotypes, metabolism, food intake, and M6A. And it seems that what we might be looking at is sort of a Goldilocks model, which traditionally I don't think we like to think about very much. We like to think that, you remove something, it goes one direction, the effect goes one direction. You add something, it goes the opposite direction. And what we have here, it seems to be that you need just the right amount of M6A. And so if you completely remove it, you get a decrease in lifespan, and maybe a too large decrease in food intake, in fat accumulation, and associated phenotypes. But if you remove just half of it, you actually have a positive effect. You have an increase in lifespan. And we know, in fact, that moderate caloric restriction seems to result in an increase in lifespan or an increase in a positive effect in metabolic phenotypes in a lot of different organisms. So we have a drug that targets METL3, which also has been shown to have promising effects on treating myeloid leukemia. And that's not our study, the leukemia one. But we want to try and see if we can replicate this effect on lifespan using a drug that targets METL3. And for all the people in the room that work with mammalian model organisms, if you then would like to try this with us in a mammalian model organism, it would be great to start a collaboration on this. Okay, so we know that food-related phenotypes are tightly linked to sleep. And in fact, many intellectual disabilities are comorbid with both eating disorders and with sleep disorders. And we know that M6A seems to affect circadian rhythms, which is one of the things that affects sleep. So we looked at sleep, and in flies, this is a relatively high throughput assay. We put them into a little glass tube that has an infrared beam going through the middle, and it just records every 30 seconds whether a fly is crossing that beam or not, so we can get an activity pattern of the fly over the days. And so we get sort of these activity actograms, and we see these so there's 3 days in a row here going from top to bottom, and we see these very typical activity patterns where flies are active in the morning. So here, this is a light diet, they're in a day-night cycle, and lights go on at 8 a.m. and go off at 8 p.m. And so after lights go on, they're active, and then they take a midday nap, and then they're very active in the afternoon again, and then they go to sleep when lights go off. So as opposed to mice, they're diurnal, like us humans, and not nocturnal. So if we look at METL3 knockout animals, you can see sort of a flattening of these very conserved patterns that you usually see in these animals. And if we quantify this, this translates not to a different total amount of sleep that METL3 mutants show, but more it translates into a fragmented sleep phenotype. So they sleep for shorter amounts of time compared to wild type or the rescue animals, but they sleep more often. So their sleep is fragmented overall during the day. And we know that sleep and non-fragmented sleep, so sort of solid blocks of sleep, are necessary to clear activity-induced ubiquitin or lysosome aggregates in the brain. So this is a study which is currently on bioarchives that I've collaborated with on during my postdoc. It doesn't involve METL3 or M6A yet, but we show that when animals don't sleep, so neuronal activity results in these ubiquitin or lysosome aggregates in the brain, which are aggregates that are responsible for protein degradation. And then during sleep, these are cleared from the brain. So when you don't get sleep, they are not cleared from the brain, and this eventually results in a deficit in neuronal function. And in fact, these ubiquitin aggregates have been shown to be linked to intellectual disability, both in fMR1, in relation to fMR1, and in relation to ZC3H14. And if we look at these METL3 mutants, if you compare control to the loss of function, and here in purple that's just the aggregates, you in fact can see a significant amount of aggregation, indicating that perhaps because the lack of sleep, there is a deficit in neuronal function because these protein aggregates can't be cleared from the brain at night. And now the actograms that I just showed, those are sort of the typical ones that we always see in the literature. But as I was reading the literature for this, I saw that depending on the study, the total amount of sleep seemed to be quite different that they reported. So we're like, okay, maybe there's some individual differences here. So we looked at a few different strains. I'm showing 4 here. And in fact, we saw that depending on what strain you look at, and so these strains are isogenic, so they have a different genetic background, each of them is isogenic, you can see that the sleeping patterns are quite different. And then I thought, well, everyone always only looks at males in this phenotype because females lay eggs, and that's a nuisance in the setup, and so people just don't do it. So I looked at females, and first I thought, well, okay, I'm doing something wrong. This is clearly not working. They don't show any of these patterns, but I did it a few times, and females, in fact, show quite different sleeping patterns than males. And I'm sorry, the sexes here are switched, so the top are in fact males and the bottom are females. So of course now we want to go back to our original results, which are in males, and see if we can replicate these in females or if they look completely different. So if we quantify this, this results in an average sleeper individual, this is percentage of the day, in male flies of about 50 to 75%, and in females of only 25 to 40, which from a Drosophila perspective makes a lot of sense to me because females have much higher metabolic requirements because they're laying and producing eggs, and so they spend a lot more time active and foraging than males do. So we are, of course, interested in where this regulation is happening in the brain. And there is a few different circuits in the Drosophila brain that we know regulate sleep and feeding behavior. And the first thing that we did was we looked at single-cell RNA-seq data of the brain to see if we could see where these genes are being expressed, to see if we could infer maybe in what circuits they're acting and where they're exercising their function. So if you're not familiar with RNA-seq data, these are sort of clustered plots, and each little cluster of cells, so every dot is a single cell, and every cluster of cells represents a group of cells that are similar to each other. And if we look at METL3, here the red cells are the one that express METL3. They're distributed quite evenly throughout the brain. There doesn't seem to be one specific brain area or brain cell type that expresses this, but more like a low level across the whole brain. If we co-localize this with a neuronal marker, and the co-localization in these plots is in yellow, you see some overlap with neuronal markers, so it's expressed in neurons, but not in all. You see some overlap with repo, which is a glial marker, so it's expressed in glia, but not in all. And then if we look at overlap between METL3, which is that core protein, but with some of the cofactors or some of the readers, again, you see some overlap in some cells, but not in all. And it's very difficult to identify a specific region where this overlap might be happening in. So we put this to the side and we're like, okay, maybe single cell RNA-seq data is not going to tell us which cells are important for this regulation at this point. But it seems more to indicate that METL3 is required in a lot of different cells, probably for a lot of different things, and that the exact function is determined by the co-expression with its different cofactors and with its different reader proteins. So the last thing I want to talk about today is how we're going to try and approach genetically how to differentiate this different effect in different cells and also in different individuals. So I'm not going to go into too much detail, but I'm very happy to talk about it after my talk if you like. But basically, the really amazing thing about flies is that you can do anything with their genome that you can come up with. So we designed a construct using CRISPR that allows us as a first step to just delete the gene. And we've done this for all the genes that we know might be involved in M6A. And at the same time as we deleted the genes, we introduced what we call a landing site. So it's a genetic construct that can be recognized by specific proteins and thus targeted afterwards. And so it's in the endogenous location of these genes that we deleted now. And then looking just quickly at these null mutants that have the genes deleted and at how viable they are, there's one interesting aspect that some of them, especially the METL3 and the METL14 mutants, seem to show some defects in viability, but a lot of the reader proteins don't show any defects in viability. So there really seems to be a lot of different pathways through which M6A is acting, and there is so much work to be done to still figure out what those pathways are and what exactly the protein interactions are that we're looking at. So the second step of this strategy, which is the one that I'm very excited about, is that we're introducing into the gene that we just deleted now a construct that has a mini gene, so basically a rescue element that reintroduces the gene. But and it is under the regulation of the endogenous promoter. But it is flanked by two genetic sites that when we express a Cre recombinase in these animals, and we can target the expression of that Cre recombinase to any cell, to any stage of development, to any tissue, then it will, in the living animal, flip this construct around, delete the gene, and express a non-functional fluorescent marker instead. So now we can take an animal, bring it through development normally, have it have a normal brain development, study its behavior as a normal wild-type animal, then express a recombinase and make that animal a mutant, and look how the behavior changes. So I think this will really allow us to, with time, get into the details about how different individuals, how different genetic backgrounds, how different sexes react to genetic manipulations, and how these pathways are involved. And yeah, so that's just a schematic. And with that, I want to thank my lab, of course, which is only just starting up, and my collaborators, and also my funding.